Hello friends, today we are starting chapter number 2, that is biological classification. In previous chapter we have gone through introduction of the biological classification and what is the reason or the importance of biological classification. Now emerging the need of biological classification is due to the importance of various types of plants and animals with respect to the evolution of humans. In early era where humans are evolving or humans were evolving, at that time we found that varieties of the plants and animals are useful to us by various means. So we started using various plants and animals. So only few plants and few animals used to be easily identifiable with respect to their use or importance in our life. For example, if I talk about some plants or some animals which are useful for us to or as a food, so that way due to the use we started identifying various plants and animals. This is a key factor, this is a root cause why we requires few plants or few species of animals to be identified. So depending on our needs, we have started them to identify, we have given them particular names, etc. So this is a root cause why we have started to classify various types of organisms. Ready? Now very first thing related to classification has been started by the scientist named Aristotle. In ancient era, first time Aristotle has started to classify various types of organisms. So the earliest to attempt, right? So Aristotle was the first to attempt the scientific basis of classification of organisms. Now what he has done, he classified plants into three groups. Number 1, tree. Number 2, shrubs. And number 3, herbs. And he classified animals into two groups, that they have red blood or not. So here you can see in the chart animal with red blood or without red blood. Again, no red blood. The animals which are not having red blood are divided into two subcategories. Either they have hard-bodied or soft-bodied. If they are red-blooded, they are mammals, lizards, birds and fish. If they don't have red blood and hard body, they are insects. If they have soft-bodied, with shell or without shell, if with shell, then it is shellfish, and if they are not having shell, then it is jellyfish. So this is the classification which was first time or earliest made by the scientist named Aristotle. So it is also known as Aristotle's classification. Now the limitation of Aristotle's classification is it is utterly based on the external character. External character is also known as morphological characters. So as they are only based on the morphological characters, external characters, it is very primary type of classification. As time have evolved and as we found the requirement, we have changed the mechanism of the classification. Ready? So please take the screenshot of the slide so that you can make your own notes. Now, as the time passes, as we need more and more, we found more and more needs of various types of organisms, we started identify more and more number of plants and more number of species. So our inquisitiveness has increased, right? Our inquisitiveness of studying them based on the three requirement. Number one, we were using them for number one, our food. Number two, for habitats to live upon, and number three, as a cloth, we started using few plants and animals. For example, the animal fur we started as a cloth in earlier life or earlier life history of humans. So we have lot of uses. So based on the uses, we have started classifying them. For example, Aristotle has started with the earlier mode of the classification. Now the changes which are made, these changes are started by the various scientists, by various mechanism, and it has been first time introduced by Linnaeus. So Linnaeus was the first scientist who gave a particular and more scientific type of classification, and he proposed two kingdom classification. He proposed the two kingdom classification is 1758, and that classifies the organism into two kingdoms, that are number one, plants, and number two, animalia. So we have major organisms and which are around us, which we can see by our naked eye, right, into two kingdoms. If they are green, they are plant kingdom, which are photosynthesizing, and if they are not preparing their own food, they are placed into kingdom animalia. So that's the first further we can say more scientific kind of classification which have been designed by Sir Linnaeus. So this classification is known as two kingdom classification, as it is consisting two kingdom plants and kingdom animals. In Latin it is known as plantae and animalia. Please take the screenshot of the slide. Now, two kingdom classification right now is not acceptable because of few drawbacks of two kingdom classifications. Now, which are them? Now, if I talk about prokaryotes, which are bacteria, cyanobacteria, or some eukaryotes like fungi, moss, fern, gymnosperm, angiosperms, all were included into a single category called plants. So plant consists from bacteria to angiosperm altogether. So it is based on only by presence of cell wall. So plants is categorized by presence of cell wall only. There is no clear-cut differentiation between prokaryotes and eukaryotes in this classification. So it is a mixture of various groups, various different kind of organisms. Number two, it is also a mixture of unicellular and multicellular altogether, ready, in a same group. For example, Chlamydomonas and Spirogyra were included under algae. So Spirogyra multicellular, Chlamydomonas unicellular, both are placed into a single group. So it is also not logical. And the last drawback what we can see is it did not differentiate between heterotrophic or autotrophic plants. Heterotrophic are fungi and autotrophs are green plant. Ready? For example, if you see the first topic, right, the fungi, moss ferns are green plant and fungi is the heterotrophic. So it is also placed under the same roof. So that is also not logical. So two kingdom classification has their own limits, as we have found different types of the plants, as we have gone through their various types of characteristic feature. We came across with the help of so many technology that we have also microorganisms, that is unicellular. We have so many multicellular organisms, but we can see only multicellular. Ready? So at that time, two kingdom classification was adequate enough to understand, but as technological advancement have been made and we identified the unicellular organisms, specifically prokaryotes and eukaryotes, when we have classified differently, then we need more advancement into two kingdom classification. So right now it is not at all acceptable, right? And we have gone through the reason why they are not acceptable. So please take the screenshot of this slide first. Right now we use the mechanism of classification which is given by R.H. Whittaker, which is given in 1969, and it is known as five kingdom classification. because it is including 5 different kingdoms. Ready? Number 1, Monera. Number 2, Protista. Number 3, Fungi. Number 4, Plantae. Number 5, Animalia. So he proposed a particular and more adequately differentiated and most scientific method of classification at that time, which included 5 kingdom, and that is why it is known as 5 kingdom classification. Now he considered few aspects to classify the organisms. Now it is based on number 1, cell structure, number 2, the thallus organization, that is body type, number 3, mode of nutrition, number 4, reproduction, and lastly phylogenetical relationship. Each of them I am going to explain you later on. So these are few basic points on which he has started classifying various types of organisms. Now, as I have already discussed, that he described organisms into 5 subcategories. Number 1 is Monera, number 2 is Protista, number 3 is Fungi, 4th is Plantae, and 5th is Animalia. So these are called 5 different categories or kingdoms, and due to these 5 kingdom, this system of classification is termed as the 5 kingdom classification. So please take the screenshot of this slide. Now let me clarify the various characters of 5 kingdom classification and reason, or the main factors on which or through which Whitaker sir has classified those organisms into various categories. So we have 5 kingdoms. Number 1, Monera, number 2, Protista, number 3, Fungi, number 4, plants, and number 5, animals. Now if I talk about those 5 kingdoms, they are basically differentiated with particular type of characteristic features like cell types, presence of cell wall or not, types of nuclear membrane, their body organization, and their mode of reproduction, mode of nutrition. So first of all, if I talk about the type of cell, if they are prokaryotes, then they are placed into category number 1, that is Monera. I repeat, if they are prokaryotic, then they are supposed to placed under the category or kingdom Monera. If they are eukaryotes, it can be Protista, fungi, plants, or animals. Now, second characteristic feature differentiating eukaryotic cell either as protista, fungi, plant or animal. That is again whether they are unicellular or multicellular. So, second important phenomena which are taken into consideration is body organization. If they are unicellular, they are protista. So, both are having cellular arrangement. If you see entire body made up of only single cell. Fine. So, here cellular and here as well they are cellular. So, prokaryote which is cellular, it is monera, and eukaryote which is having cellular organization, means entire body is made up of cell, they are protista. Now, all the multicellulars, how they are differentiated into fungi, plant or animals? It is defined with most number of the categories called either presence of cell wall or mode of nutrition. Now, if cell wall is not present, I repeat, if cell wall is not present, they are animals. If cell wall is present but not cellulose, they are fungi, and if cellulose, they are plants. Mode of nutrition, though they have cell wall, but their mode of nutrition is heterotrophic, specifically saprophytic or parasitic, they are fungi, and if they are autotroph, then they are plants, and if they are heterotrophic, specifically holozoic or saprophytic, they can be animals. Now, body organization, multicellular comprising loose tissue, they are fungi. Multicellular have organs, here it is tissue, here it is organ, they are eukaryotic. And organism from tissue to organ systems, they are animals. It does not mean animal do not show tissue level organism. There are few animals which are showing tissue level organism, there are few animals which are having organ level organisms, ready, or majority of them are organ system level of the organism. Even plants, some are having tissue level organism and some are, most of them are having organ level of organization. So, this level of organization we have already covered in chapter number 1 while describing in the very first lecture. So, these are the basis of the classification named 5 kingdom classification. Ready? So, these are the few aspects which has been considered by R.H. Whittaker to classify the organisms into 5 kingdoms. Now, if I talk about the last category of the characteristic feature is nuclear membrane. The membrane around nucleus, in Monera they are not found, but in all the eukaryotes they are found. On base of the nuclear membrane only we are classifying the cell either as prokaryote or as eukaryote. So that is also to be considered, whether they are having the nuclear membrane, then they are eukaryotic, and if they don't have nuclear membrane, they are prokaryotic. So cell type is defined with the presence of nuclear membrane or not. If nuclear membrane is present, then the cell is considered as eukaryote, and if nuclear membrane is not found, then that cell is considered as prokaryotic cell, as we know. So that's the story of 5 kingdom classification, and please take the screenshot. Now comes the next level of the organization, or next level of the classification, called 3-domain system. Now more advancement in 5 kingdom classification. Now this classification mechanism is proposed by Carl Woese. Now, as we know that we have 5 kingdoms. Number 1, Monera. Number 2, Protista. Number 3, the fungi. Number 4, the plants, and number 5, the animals. Now first, Monera is more closely studied and further divided into 3 subcategories. Entire mechanism has been divided. So major work is done into the Monera, and then the entire 5 kingdoms has been rearranged into 3 domain. Ready? So this mechanism is known as 3-domain classification. Ready? If you see on the top, it is termed as 3-domain classification or 3-domain system, which is proposed by Carl Woese. Now what is this domain? Domain number 1 is Eubacteria, domain number 2 is Archaebacteria, and domain number 3, what he termed, is Eukarya. Now actually, according to the feature, this must be first Archaea, then 2 Eubacteria. Now how he has classified them? He involved evolutionary relationship into them. Evolutionary relationship he added. Now how it is affecting to that? For example, if I am talking about the early life, which is first time evolved onto the earth, at that time earth was at all not suitable for condition of sustaining of the life. Conditions on the earth were very harsh. Atmosphere around the earth was very, very hot. The earthquakes, right, and very oftenly the calamities are continuously going on. So many different kind of calamities. So at all it is not suiting to the life. Now the water reservoir, for example, if I am talking about oceans, they are filled with the water which is very hot. The pH was unbalanced. Ready? Maybe with highly salty, maybe highly hot, maybe acidic water. So in those harsh conditions, first time life has been arise. So in harsh condition, first ever monerans have started to evolve. So he considered that mechanism as well. So if they are adapted to this harsh condition, they are different than modern monerans. So modern monerans are eubacteria, And archaebacteria are the oldest primary organisms which are evolved, which are further subcategorized into number 1 methanogens, number 2 halophils, and number 3 thermoacidophils. Ready? Now, what are methanogens? By their own mechanism of metabolism, they produce methane gas. That is why they are said to be methanogens. Ready? So these are few bacteria which are found in gut region of few special type of mammals. Ready? Second, halophils. They are found into the water with high salt. It is halophils, where high condition of the salt is there. There they are found. And the last one, third category, is thermoacidophils. If the water is with high temperature or low pH, that is acidic water, they are found in those kind of extreme conditions. So they are actually naturally found in extreme conditions, not in a normal condition. So these are few bacteria which are found into abnormal condition or at the condition where normally life is not possible. So basic characteristic of those organisms are they have circular DNA, they are prokaryotic, their cell walls do not have peptidoglycan, they have branched chain of phospholipids in cell membrane, they are antibiotic insensitive, they do not harmed by the antibiotic, and methionine for first AA. You cannot be able to understand this right now, so I am not going to explain it. As far as eubacteria is concerned, they are the modern bacteria which are evolved by course of the time later on onto the earth, and they are furthermore classified based on their characteristic feature of the cell wall. Now, one special mode of observing them under the microscope is by using various types of stains. So, if any cell has to be observed under the microscope, we are supposed to use this stain. So, Gram stain is prepared by the scientist called Gram, and it is called Gram stain. So, if they are stained by these Gram stain, then it is called Gram positive bacteria, or if they are not, then it is Gram negative bacteria. Few are spirochetes and few are cyanobacteria. So they are in 4 categories. So Gram positive, Gram negative, spirochetes and cyanobacteria are 4 basic types of eubacteria. Gram positive, they are mostly harmful bacteria which are causing various types of disease. Gram negative, very useful bacteria. For example, when we are talking about making of curd, making of wine, etc. So they are useful for us, with specially they are causing the fermentation. So useful bacteria, Gram negative. Spirochetes, again pathogens, and cyanobacteria, they are photosynthesizing bacteria, special types of bacteria photosynthesizing. So these are different categories of eubacteria, and the common most characteristic features are circular DNA, prokaryotic cell wall, but cell wall do not contain peptidoglycan and they have straight chain phospholipid in their cell membrane. They may be antibiotic sensitive and they have F-formyl methionine for first amino acid. Ready? Now rest of all the four categories protista, this is classification or this is division of only Monera. Monera are divided into first Archaea and second Eubacteria. Rest of all, that is Protista, Animalia, fungi and plantae, they all are categorized under Eukarya, that consist eukaryotic cells. They have linear DNA, they are eukaryotic cells, cell wall variable if they are present, different kind of cell wall can be present, straight chain phospholipids, antibiotic insensitive and methionine for first amino acids. So these are the categories what we have understood with reference to 3 domain classification which has been given by Carl Woese. So we will move further into the next lecture, and I am stopping my discussion right now. I'll see you into the next lecture. Please take the screenshot. Take care and have a nice time.